let's look at question number 2 from GATE Biotechnology 2018 paper. So the question is, if serum is removed from the growth medium of human embryonic kidney cell line HEK, then the cells will option A proliferate faster, option B proliferate normally, option C undergo cell cycle arrest, and option D undergo immediate apoptosis. Now before that, what is serum? So the clear liquid that can be separated from the clotted blood is the serum. And blood serum is about 90% water with dissolved proteins, minerals, hormones, and carbon dioxide, and is an important source of electrolytes. So in the blood, the serum is the component that is neither the blood cell, that is, serum does not contain the white or red blood cells, and it does not contain a clotting factor, but it is the blood plasma with fibrinogens removed. So serum is basically the plasma minus the clotting factors. So like we just saw, serum is a complex mixture of albumins, the proteins, growth factors, and growth inhibitors. So serum is one of the most important components of cell culture media and serves as a source for amino acids, proteins, vitamins, particularly the fat soluble vitamins like A, D, E, and K, and carbohydrates, lipids, hormones, growth factors, minerals, and trace elements. So serum, especially the fetal bovine serum FBS, is commonly added to cell culture media as a supplement because it contains all these essential components which we just saw. So these components provide signals for cells to progress through the cell cycle and carry out various cellular functions. So it's basically like feeding the cells with all the nutrients. So the normal growth media contains 2 to 10% of the serum. Now, what are the effects of serum being removed from the media on the cultured cells? So when serum is removed from the growth medium of human embryonic kidney cell lines, the cells may experience a reduction in the availability of the essential nutrients. So as a result, several cellular responses can occur. So the major one is cell cycle arrest. The cell cycle is a highly regulated process, and cells require specific signals to progress through the phases of the cell cycle. So in the absence of these signals that are provided by the serum components, the cells may undergo cell cycle arrest, halting the progression at a specific phase. So this can lead to a decrease in cell proliferation. And then quiescence. The cells may enter a quiescent state, which is a reversible non-dividing state. So in the absence of proliferative signals, the cells can temporarily stop dividing until the appropriate signals are reintroduced again in the media. And then changes in gene expression. So removal of serum can lead to changes in gene expression patterns such as the cells respond to altered environments. So some genes associated with cell cycle progression may be downregulated. And then apoptosis. So while immediate apoptosis may not always be the primary response when the serum is removed, it can happen over time if the cells are unable to adapt to new conditions and if the lack of essential signals triggers apoptotic pathways. So it's important to note that the specific response may vary between different cell types and experimental conditions. So researchers often manipulate serum concentrations in cell culture experiments to study cellular responses and mimic conditions relevant to specific research questions. But the overall idea is that when serum is removed, the cells stop proliferating because the cell cycle is arrested. So now coming back to our question, if serum was removed from the growth medium of HEK, then the cells will, we definitely know that the proliferation reduces. So they do not proliferate faster and they do not proliferate normally. They do undergo cell cycle arrest, which answers the question. And we just saw that they might not undergo immediate apoptosis. They undergo apoptosis later when the cells respond and they don't get adapted to the new environment. So yeah, if the serum is removed from the growth medium, they undergo cell cycle arrest. The cells will undergo cell cycle arrest.